Well, thank you all for hosting me today. It's a real pleasure to be here. And Simon, look forward to talking to you a bit more about medical school over dinner. So I'm going to talk about work we've been doing here at the CRUK on a study called TRACERx. I'll explain about that in a second or two. But I just want to sort of revisit the lectures on cancer that Simon and I sat through at QMUL all those years ago at the tender age of 22. And we were taught that cancer evolves in a linear manner, or at least I was taught. I'm not sure Simon turned up to those lectures. What we see is sort of linear evolution with selective sweeps occurring at the onset of each new oncogenic or tumor suppressor event that occurs during tumor evolution. And when you Google cancer images and you look at the drop-down menu, you see images like this with a homogeneity of tumors and linear avogogram-like models. And yet, there was plenty of evidence in the literature, or at least opinion pieces in the literature from people like Gloria Hepner and Peter Knoll and others that have put forward this idea in the 70s that cancer evolves in a branched manner. And I guess it seems utterly obvious to us now. But this is something we wanted to test back in 2008, 2009, when we got a small grant from the European Union to study cancer evolution and drug resistance in kidney cancer. And this was just sort of the advent of whole exome sequencing. And we took multiple biopsies from this early stage renal cancer with a large collaboration at the time with my colleagues and friends, James Larkin and others at the Marsden, Zoltan Selassie and our bioinformaticians at the CRIK. And we took multiple biopsies and just asked a very simple question. If you take one biopsy for a biomarker study, personalised medicine, as it was known then, as it's still known now, how reliable is that biopsy in giving some sort of picture of the genomic landscape of the disease when subjecting that biopsy to whole exome sequencing? In the top right-hand corner, you can see the number of coding mutations that we found in every biopsy, and the number of coding mutations found in some biopsies and not others, depending on whether they're in the primary or the metastasis or specific to individual biopsies. And of course, you can reconstruct crude phylogenetic evolutionary histories by finding mutations present in every tumor cell, in every tumor region, VHL being the canonical tumor suppressor in clear cell carcinoma of the kidney. And tumor suppressor events that are found in some parts of the tumor and not others. SETD2, KDM5C, and others. And you can see here that SETD2 is inactivated in 3 different ways in 3 different parts of this patient's tumor. And I'll come back to that in a minute, because it gets to the sort of heart of what we're interested in, which is sort of these evolutionary rule books that constrain tumor evolution, that ultimately I hope there may be some utopian vision of being able to predict the next evolutionary move a tumor may make. Now, of course, as Peter Knoll said in his 1976 paper in Science, his editorial on how he felt that cancers evolve, I mean, this guy was a true hero and never sought any publicity at all, and died about 18 months ago. He really recognized the sort of Darwinian nature of tumor evolution, and really hasn't ever been properly recognized in the field, sadly. And of course, Charles Darwin had this all wrapped up nearly 200 years ago in his well-established theories of evolution and natural selection. And of course, this all plays out in cancer as we know it today, of cell-to-cell variation, which essentially acts, is essentially the substrate upon which natural selection acts, and pans out in the sort of regional diversity of cancer subclones that we're more familiar with now. And essentially this intrinsic problem of each patient having a unique phylogenetic tree. Phylogenetic tree, that means you have tumor clonal alterations, clonal driver mutations, and a panoply of subclonal driver alterations that may be unique from patient to patient. And I think these things have an important impact on our ability to cure metastatic disease, arguably, the cost of cancer drug development, and our ability to qualify biomarkers for clinical use. So with those early data in mind, we set up these 2 longitudinal cancer programs called TRACERx, one in kidney cancer, which I won't talk about today, led by my friend and colleague Samra Chiralec, who's a clinician scientist based in my lab and also a medical oncologist at the Marsden, and a thoracic medical oncologist and lead for lung cancer TRACERx, Mariam Jamal Hanjani, two very talented clinician scientists who really have been absolutely amazing in leading these two studies. And the idea of these studies is that if we extensively map and sample the primary tumour and the metastatic sites ultimately over the disease course, we'll ultimately, we hope, begin to distinguish metastatic competent from incompetent tumour subclones in the tumour and start to be able to infer mechanisms of branched evolution, the relationships of tumour heterogeneity to clinical outcome, whether there are specific evolutionary rule books that we might be able to define and ultimately predict, whether there are clear timing of driver events during tumour evolution that we might be able to guess the next evolutionary move a tumour may make and forestall it. Can we identify the origins of the lethal metastatic subclones in the primary tumour and infer mechanisms of branched evolution and immune escape? And it's really the latter that I'll be talking about today, mechanisms of that foster cell-to-cell variation and ultimately, we think, impact upon immune escape. Now, in evolutionary history, you'll be familiar with the sort of two opposing schools of thought in evolutionary biology. There's microevolution, which is essentially an extension of Darwinian evolution. That is, small mutations that occur over a lengthy period of time that lead to profound phenotypic change over a very prolonged period. So small incremental steps make large changes over a lengthy evolutionary time. And in the context of lung cancer evolution, that can be thought of in terms of these three specific mutational signatures. You have a tobacco C-to-A carcinogen signature, mutational signatures you'll be familiar with. The APOBEC cytidine deaminase mutational signature. These are C-to-T mutations at TPC dinucleotide sites. And then the spontaneous deamination signature, these C-to-T mutations. These are the three common mutational signatures seen in lung cancer evolution. So we were interested in asking, when do these mutations occur? Do they occur early or late or a mixture of both? Is it just a mess? And what we see in the context of lung cancer evolution, bear in mind, this is an NHS cohort of patients coming through clinic with early stage non-small cell lung cancer. So about 95% of these patients are ex-smokers or current smokers. What we find is that the dominant mutational signature in the trunks of these tumours' evolutionary trees, so these are the mutations present in every tumour cell, are C-to-A smoking mutations. And they dominate the trunk of the tumour's evolutionary tree. You can see in grey here, you can see the large panoply of grey C-to-A mutations. Now, the intriguing thing is that we also in TRACERx acquire smoking histories from all of our patients in terms of PAC years. And in lung adenocarcinoma, but not lung squamous carcinoma, the number of PAC years that patient's been exposed to directly correlates with the length of the tumour phylogenetic tree in terms of the number of C-to-A mutations, which is quite remarkable in many ways. So I think it sort of speaks to, first of all, obviously the honesty of patients and their ability to sort of report smoking histories and how reliable they are, something I was quite sceptical about, but also the mutagenic impact of tobacco exposure over the life history of that particular tumour. So what about the branch mutations? Well, interestingly, the branch mutations are very different. The branch mutations now, we no longer see a significant association between PAC year exposure and branch mutations, and maybe that's not that surprising because as the tumour grows, it's probably growing away from the airways and it's no longer exposed to tobacco in quite the concentrations it was exposed to before. But now what we see is that the APOBEC cytidine deaminase signature and the mitotic clock signatures become dominant in the branches. So what this means is that if a patient presents in clinic to us who's a smoker, current smoker, actually the dominant mutagenic process is not going to be tobacco exposure. It's going to be two endogenous processes, this mitotic clock signature and the APOBEC cytidine deaminase signature. So of course the question that we keep asking ourselves is why? Because if this was not advantageous ultimately, if this was not advantageous, the tumour genome would have found a way of shutting APOBEC down. Forgive me for that anthropomorphic vision, but because I've only got about 45 minutes, I will occasionally slip into that view. It's wrong. There's selection and diversity. And what's happening here is that for reasons we don't fully understand yet, APOBEC is selected during tumour evolution. The APOBEC cytidine deaminase signature is selected during tumour evolution. So we've turned to HIV to try to understand why this might be, and of course the virologists have been thinking about this for many more years than we have. And what they've found, Ruben Harris and colleagues published this paper in 2008, that inferred that there was a sort of optimal level of APOBEC3G mutagenesis that allowed HIV, or was permissive for HIV, to escape HLA recognition. And so when particular peptides from HIV are recognized by HLA and loaded onto HLA molecules, potentially APOBEC3G activity provides a means of immune escape. And too much APOBEC3G activity and the virus dies, and too little APOBEC3G activity and there's not sufficient diversification for evolutionary fitness. So we've got a small grant with CIK that's beginning to explore the relationships between HIV evolution and lung cancer evolution. That's sort of work in progress. So this is a precision medicine talk, and I just wanted to remind you and remind me to say that each tumour phylogenetic tree is different. So ultimately, thinking about precision medicine in medical oncology is not a trivial task at all. And there are unique sets of driver events, both clonal and subclonal, for each patient. And of course, the genome instability processes and mutational signature processes differ in distinct branches of each individual tumour. So when thinking about targeting genome instability processes, that's a pretty tricky task as well. So APOBEC, we think, is a major driver of cell-to-cell variation and branched evolution. So I mentioned two opposing views of evolution in evolutionary biology over the last, I guess, century or so. And the second opposing evolutionary view is pioneered by this chap called Goldschmidt, who was an evolutionary biologist in the 1940s, who published a book called Material Basis for Evolution that was rather controversial in its time and remains so today in some ways, because he made the seminal mistake of wrecking his career by telling everybody that Charles Darwin was wrong, and saying that essentially evolution plays out through the rearrangement of the serial, what he called the serial chemical constituents of the chromosome, into a new spatially different order. So he postulated that chromosomal instability was the primary driver of speciation, rather than small incremental change over a very long period of time. He said actually just occasionally you get this rapid diversification of the genome into a new spatially different order with rearrangements of the chromosomes that ultimately leads to a new species. Now, he may or may not have been right about that, and I'm skeptical that he was right, although I'm going to show you a slide in a minute that suggests he may well have been, in terms of speciation. But I think in terms of cancer evolution, he was really spot on. And in late-stage disease and in even early-stage disease, we see increasing evidence for this large-scale rearrangement of what he would say the serial chemical constituents of the chromosome into a new spatially different order. That's exactly what we see in cancer biology across most intractable solid tumours. That is rearrangements of whole chromosomes, parts of chromosomes, in number and structure across multiple cells across an evolving tumour mass. Now, if you think about it, a small 10 centimetre cube tumour, non-small cell lung cancer, stage 1 disease, will have 300 million tumour cells. And when we start to image these tumour cells under the microscope, it's not unusual in every cell division to see a chromosome segregation error. One can now imagine that if you're gaining or losing a chromosome every cell division in 300 million cells across a very small tumour mass, the capacity for diversification is really almost unfathomable. And when thinking about speciation, there was this paper published in 2016 focusing on the gibbon. And the gibbon diverged from other common apes about 4 million years ago. And unlike other common apes like chimps and gorillas that have 24 pairs of chromosomes, gibbons have between 19 and 26 pairs. And they're very different from many apes in that they walk upright and they don't have a tail. and like us, or many of us, they're monogamous and operatic sopranos. But interestingly, unlike us, that we have this 8-boned carpal wrist, they have a ball and socket joint for their wrist. Very different from all other apes, and that enables them to swing from branches at nearly 60 kilometers per hour. They've got an incredibly mobile, strong wrist. Now, when Carbone and colleagues sequenced the gibbon genome, what they found is these retroviral mobile DNA elements that had inserted into the genome, and through molecular archaeology wizardry, they reckon that these mobile elements had inserted round about the time the gibbon diverged from other ape species. And the intriguing thing about these mobile elements is they think they disrupted genes involved in chromosome segregation, spindle assembly checkpoint genes like BARBAR1, MAD1-like 1, and kinesins such as KIF27. And then when they looked at these genomes, they saw there was very rapid evolution around these genes involved in chromosome segregation, suggesting that perhaps there'd been rapid diversification of the karyotype, and then this sort of selective pressure to restabilize the genome into a new spatially different order. And perhaps that's what they think led to this very unusual karyotype of the gibbon genome, and potentially this very rapid evolution and these profound differences between the gibbon and other species. Now, why am I talking about this? Well, I'm talking about it because I think it's important in cancer. Because I think if you can see that you can get whole new species evolving through chromosomal rearrangements, one can begin to see the scale of the problem we're dealing with in cancer evolution when we're dealing with cell-to-cell variation at the chromosomal level in a very small tumour containing 300 million tumour cells. And when one thinks about dealing with patients who have advanced disease, when one's dealing with potentially 1 trillion or more tumour cells, one can begin to imagine the complexities inherent in cancer evolution in trying to deal with a scale of chromosomal rearrangements that might be working in that tumour and engaging and preventing us from successfully curing a patient. So when we look at TRACERx, we actually find that chromosomal instability is very closely associated with outcome, at least in the first 100 patients. We find that tumours that are more heterogeneous from a chromosomal karyotype perspective have a worse clinical outcome than tumours that are much more chromosomally stable. Now, this is data from only the first 100 patients. It's very preliminary. We'll have much more data in the context of the next 6 to 12 months. But so far, it seems that in multivariate analysis, chromosomal instability is overwhelmingly associated with poor outcome independent of disease stage. And Goldschmidt said in his book, Material-Based Evolution, that these mutations, these large-scale evolutionary events, need only be of the most extraordinary rarity to provide the world with an important material for evolution. So that one chromosome segregation error may be sufficient to drive diversification and evolution. So we're very interested in understanding whether this is important or not. And I still sit on grant committees where people maintain that chromosomal instability is just a bystander effect of replication stress, or other aberrations in mitotic signaling, mitotic control, and what have you, and is not at all important in diversification and selection and evolution. And I want to try to show you some early data that may convince you otherwise. So first of all, how does this all come about? And one of the ways we think it comes about is through genome doubling. So what we find in TRACERx is that genome doubling, which is exactly what it says it is, a doubling of the whole genome, occurs very commonly in lung cancer, and in about 3/4 of cases we can find evidence of genome doubling in the cancer genome. And when it occurs very early on in tumour evolution, in the trunk of the tumour's evolutionary tree. David Pellman and colleagues showed about 15 years ago now that when you model genome doubling in the lab, in a mouse model, genome doubled mouse mammary epithelial cells, mouse mammary epithelial fibroblasts, are very much more aggressive than their non-genome double counterparts. And what we find in TRACERx that the degree of chromosomal instability is very much higher in TRACERx tumours that have doubled their genome compared to those that haven't. They're very much more chromosomally complex. So genome doubling seems to be associated with chromosomal complexity and chromosomal diversity, this rearrangement, if you like, of the serial chemical constituents of the chromosome, as Goldschmidt would put it. So we were interested in trying to understand why this was. So about 4 years ago, Sally Dewhurst set up a model experiment in the lab where she took diploid HCT116 colorectal cancer cells that we notice on FACS plots have a very small population of genome double cells, but they never grow very much greater than about 2% of the population at any one time. They're always staying at roughly 2%, which suggests they're being constantly outcompeted by diploid cells. So what she did, she single-cell cloned diploid cells. She labeled their chromosomes with GFP, H2B GFP, to enable us to track chromosome segregation errors. And then over time, she noticed that in those single cells, one or two of those wells would become tetraploid. And then she single-cell cloned the tetraploid cells that had been spawned off those diploid parental progenitor cells. And she then began to track their chromosome segregation errors and, more importantly, the fate of the daughter cells over time, with a hypothesis being that, for reasons we don't fully understand, tetraploid cells can propagate aneuploid progeny, but diploid cells can't. And what we see is exactly that when we track diploid cells, if you look here, after diploid cells make a chromosome segregation error, the daughter cells following that segregation error event, only about half of those daughter cells go on to perform a subsequent mitosis within the 72 hours of the experiment. In contrast, the tetraploid cells, the genome double cells that have been spawned from those diploid parental cells, they don't seem to mind when they have a segregation error. They quite happily go on to perform another cell division, despite the fact that they've undertaken a chromosome segregation error, suggesting that, for reasons we don't fully understand, that tetraploid cells have lost that sensing mechanism that can actually sense a diploid karyotype and can sense chromosome segregation errors. And we're trying to work out in the lab what those sensing mechanisms are. But that aside, what we did is we tracked these cells over 18 months in the lab to ask, do they really become more chromosomally unstable over time? And that's exactly what we see. So the diploid cell clones over 18 months in culture, so these are 4 different diploid cell clones, 3 of which were grown up over 75 weeks in culture, and they were subject to SNP array analysis to look at their chromosomal instability indices, the degree of karyotypic variation. And they're robustly chromosomally stable. In contrast, the tetraploid progenitors from these diploid parental clones become progressively more chromosomally unstable over the course of the experiment, over 18 months in culture. And Sally showed that in colorectal cancer, the acquisition of genome doubling in early stage disease is a very poor prognostic sign, it seems. So genome doubling, we think, is associated with the ability to lose the sensing mechanism for karyotypic instability, to propagate aneuploid progenies, and essentially allow diversity to ensue. And this isn't really a new observation, in fact, that Julian Huxley in 1942, despite his gross, or rather grotesque views on racial biology and what have you, he did actually have a few sensible things to say about evolution. And one thing he said was that tetraploid forms are in many cases more generally vigorous, and the fact that's reflected in their distribution, which is frequently wider than that of the diploid variety. So I look at this and I think to myself, could this be a metastasizing tumor cell from a diploid colorectal cancer that's now doubled its genome and now can distribute itself more widely than the diploid variety? Of course, that's work in progress, but we are seeing across multiple tumor types now, genome doubling is a high-risk feature and predictive of poor recurrence-free survival. So genome doubling, we think, is a driver of branched evolution. So using genome doubling, we can start to get towards an evolutionary rule book, if you like, because we can start to time events in terms of whether they're early or late. And in brief, genetic events that we're used to targeting, like epidermal growth factor receptor, are early events that occur before genome doubling. And then later on in evolution, as we're getting the APOBEC mutagenic signatures, what we're beginning to see is selection for mutations in driver genes that we think are monitoring genomic integrity or involved in DNA repair or histone and chromatin modification. And early work in the lab is beginning to suggest that some of these genes themselves, when inactivated, drive further diversification that suggests perhaps the tantalizing idea that diversity is a selectable phenotype during evolution of early stage lung cancer. So I've begun to show you some ideas about how chromosomal instability occurs, and there are many other ideas in the field that have been published well before us, but that's just sort of one point that I wanted to make, that genome doubling is important. But now I want to sort of get to the heart of why we think chromosomal instability is important. And the first thing is we think that chromosomal instability is a driver for parallel evolution. So Tom Watkins in the lab is a talented MBPhD student. He set up this pipeline which enables us to haplotype heterozygous SNPs using multi-region sequencing data and ask a very simple question. If you see an oncogenic event in a gene such as HER2, has it occurred once or on multiple occasions during tumor evolution? And by using this tool that was published last year, we can now see that again and again and again we see evidence where, for instance, if you take CIK001 here, CDK4 is amplified on 3 separate occasions in 3 different subclones from this patient's tumour in 3 different regions of the tumour. So, as Darwin would say, the common rule throughout nature is infinite diversity of structure, in this case chromosomal structure for gaining the same end, where we're seeing amplification events being selected for multiple times from either maternal or the paternal chromosome on more than one occasion during tumour evolution. And bearing in mind that we're still very much undersampling these tumours, we're perhaps sampling less than 1% of the tumour exome, or the number of tumour clones in that patient's tumour. This is really probably just the tip of the iceberg in terms of the degree of selection that's occurring. So what about immune evasion, the sort of subtitle of this talk? So I want to show you some data that's published, and probably half of this talk will be unpublished data that's a bit rough around the edges, so you'll forgive me if it's deeply flawed, but I'd certainly appreciate your feedback. So I'll show you some published data of how we think chromosome instability is driving immune evasion. Nikki and Crispin and Rachel developed this pipeline called LOLA, loss of heterozygosity for HLA, which attempts to use cancer exome sequencing data to infer maternal or paternal loss of HLA alleles, class 1 HLA alleles during tumour evolution. And of course these are important because class 1 HLA alleles present mutated tumour peptides generated from the mutanome loaded onto class 1 MHC in the endoplasmic reticulum onto the cell surface. They're then presented to CD8 cytotoxic T cells that hopefully recognize them through the T cell receptor. And it's been known for many, many years. In fact, I did an undergraduate project through immunohistochemistry looking at HLA loss in lung cancer and didn't get very far. But I think with now cancer exome sequencing data, we can begin to infer a quite deep resolution when this occurs. And if you're interested, I can tell you how they did this at the end of the talk. And this LOLA pipeline now can infer the extent and frequency of HLA loss of heterozygosity in cancer clones and subclones during tumour evolution. And previously, one of the commonest causes of immune evasion was thought to be mutations of beta-2 microglobulin, beta-2M loss, or HLA mutations. And when we look at that, that's actually quite uncommon in untreated lung cancer. About 4% of our first 100 patients have either got mutations in beta-2M, beta-2 microglobulin, or mutations in class 1 HLA. In contrast, mutations in class 1 HLA. In contrast, HLA loss of heterozygosity is very much more common. We see this in nearly 40% of patients. And when it occurs, surprisingly, it's commonly occurring as a subclonal or branched event in some regions of the tumour, some cells of the tumour, but not others, for reasons that we're still very uncertain about and we don't fully understand. And so here's some examples where HLA loss is a truncal event occurring very early on in tumour evolution. And here's some examples, I'm sorry, it's quite small, but I'll zoom in on them in a minute or two, where HLA loss of heterozygosity is a later event occurring in the branches. And here's some very interesting examples where we see parallel evolution of HLA haplotype loss that's occurring on more than one occasion in different subclones. And they're zoomed in here. You can see the same haplotypes are lost recurrently in different subclones, different regions of the tumour, which suggests there is sort of profound selection for recurrent loss of the same HLA alleles, perhaps because there is a potent neo-antigen that's binding to those HLA alleles that enables these subclones to be selected during evolution. So you can see the loss of 6P occurring here as the sort of focal peak of subclonal loss in squamous carcinoma of the lung. So using the deep sequencing multi-region data of TRACERx, we can start to ask some, I think, quite important questions. And important because, you know, what we've been longing to do is study evolution in a patient, cancer evolution in a patient, to really understand this process. And And in a sense, because these tumour biopsies are fairly close by and that we hope that the tumour microenvironments are relatively similar, or I'll show you some data in a minute or two that may contradict that statement, and of course the patient's own germline is the same, we can start to ask, with different subclones in the same tumour, if one subclone has lost HLA and the other hasn't, adjacent to it, is there a difference in the evolving subclones distal to that maternal subclone in terms of the mutational repertoire or the neoantigens that particular subclone may hold and encode? And that's exactly what we find, actually, that in daughter subclones from subclones that have lost HLA, we see more mutations, more coding mutations, than for adjacent subclones that haven't lost HLA. Suggesting that loss of HLA is permissive for ongoing branched evolution and, if you like, evasion of the immune system by stealth. And given the relatively crude relationship between coding mutations and neoantigens, and I stress these tools are still in their infancy and crude, and you must take them with a pinch of salt using tools such as NetMHC, PAN and others, that we're finding predicted neoantigens that are predicted to bind to the lost HLA allele with high affinity are enriched in subclones distal to the HLA loss event compared to subclones that haven't lost HLA. And we think in the context of a sort of brave new world of vaccine therapies, peptide vaccines, RNA vaccines in cancer, and I hope the future of adoptive T-cell therapies, and I have got a conflict here because I've got a small interest in a rounding error in terms of stock, but I'm a founder of a small biotech company called Achilles that's aiming to look at T-cell therapies funded by Crick and the Wellcome Trust and CRUK, that's aiming to look at T-cell therapies targeting truncal neoantigens that I'll talk about in a minute or two, that we do need to understand whether the HLA alleles are still there in the tumour to be able to make sure that these mutations are efficiently presented to the immune system such that the T cells can recognize the tumour cells that are ultimately bearing those mutated peptides. So we were asked by the referees, in tumour regions that have lost HLA, do you see a significant impact in terms of the immune repertoire? Are they immune hot? Are they immune cold? What are you seeing? And what we see is that these tumours tend to be immune hot. They're immune replete. Some of these regions are packed full of T cells. That they have high expression of PD-L1 on both immune cells and tumour cells. And the question is this chicken or the egg? Have they lost HLA and has that provided a signal for immune infiltration, or has the immune infiltration selected for HLA loss? I suspect it's a mixture of both of them, actually, rather than one or the other. So if you quantify immune infiltrate in tumours with HLA LOH using RNAseq data, we see a similar thing that in lung adenocarcinoma, but not lung squamous carcinoma, interestingly, we see evidence of immune infiltrate in tumours with HLA or tumour regions with loss of HLA, including an increase in CD8 cells, increase in PD-L1 and CTLA-4, as well as potentially Tregs and MDSCs, macrophages, neutrophils, and natural killer cells, and what have you. So I'll come back to that in a minute or two in future work. And we validated this in the Cancer Genome Atlas, non-small cell lung cancer cohorts of RNA-seq data. Again, we see this in adenocarcinoma, that adenocarcinomas with loss of HLA are enriched for CD8 T cells, as well as macrophages, neutrophils, and NK cells, but not squamous carcinoma of the lung for reasons we don't understand. So we're beginning to see some evidence that maybe there's also selection for HLA loss at the primary metastatic transition. So what we're seeing here, and this is not significant, but it's only a trend at the moment because I think we're underpowered, but I'm hoping that over the course of the next 6 to 12 months that with TRACERx samples where we've now got about 300 metastatic sites, we'll be able to improve the power for this analysis. But we're beginning to see a trend for loss of HLA in kidney cancer and lung cancer between the primary and the metastatic sites, where we're seeing potential for enrichment of HLA loss of heterozygosity or selection for subclones that have lost HLA at the primary metastasis transition. We think that HLA loss of heterozygosity is maybe a permissive event for branch evolution. So losing HLA now allows cells to evolve below the radar, if you like, develop mutations that now cannot be seen by the immune system. So what are we going to do with this huge diversity? APOBEC mutations, smoking mutations, mitotic clock mutations, generating cell-to-cell variation, as well as chromosomal instabilities. I mean, the capacity for diversity is really quite bewildering. We've been successfully, at least in non-smokers, targeting epidermal growth factor receptor alterations that we've shown in TRACERx are clonal, present in every tumour cell. But resistance is inevitable, anywhere between 6 and 18 months. By and large, most patients will have developed resistance to first-line EGFR tyrosine kinase inhibitor therapies due to the selective outgrowth of a pre-existing clone that harbours resistance, a resistant, say, T790M mutation, or a subclone that acquires that resistance event over the course of disease. So what we want to be doing, the evolutionary biologists tell us, is we need to be targeting multiple clonal alterations present in every tumour cell. And we are told that number has to be 4 or more. I don't quite know why it has to be 4 or more, but I think that the calculations are essentially based on the degree of genomic instability and the population size at presentation. Ideally, we need to be targeting as many as we can possibly target to really force the tumour down a cul-de-sac, to really prevent the ability of a single cell out of a trillion to have a resistance event. And we're hoping that there's sort of a tantalizing light at the end of the tunnel, in that in the context of TRACERx, what we're seeing is that smoking lung cancer genomes that have been exposed to tobacco, either now or in the past, have about a 5 to 10-fold greater burden of clonal alterations, clonal coding mutations present in every tumour cell, compared to patients who have never smoked. And that presents us with an opportunity because, of course, if you've got 5 to 10 times more coding alterations, we're talking about tumours with up to 1,000 coding mutations in the trunk of the tumour's evolutionary tree. There's a possibility that one or more of those mutations, hopefully many more than that, will be recognised by the immune system as being foreign, and there may be immune cells in the patient's tumour or blood that can recognise those clonal mutations that we could identify and potentially expand and reinfuse back into the patient. So the first question is, are these clonal trunk mutations important? And this is a validation cohort of a paper that Nikki and our lab published, Nikki McGranahan and our lab published in 2016, that alludes to the fact that tumours with a very high clonal neoantigen burden, that is tumours with very long trunks, packed full of coding alterations, in the upper quartile of coding alterations in the TCGA, do very well. And so we've validated this now in TRACERx, that these tumours in the upper quartile of clonal neoantigen burden have a very good disease-free survival outcome and very rarely relapse after adjuvant chemotherapy or after surgery plus or minus adjuvant chemotherapy. In contrast, subclonal or branched neoantigens don't seem to make very much difference to outcome. And in fact, this is a small cohort of only about 100 patients. But Ellie Van Allen's group at Dana-Farber have recently shown actually subclonal neoantigens may be bad, at least in terms of immune response. And I'd love your opinion as to why this might be. My view on it may be that perhaps the gross diversification of the tumour mass distracts the immune system from what's really important, crudely as a non-immunologist, which is the truncal alterations present in every tumour cell. So what we think is, our hypothesis at the moment is that tumours with very long trunks and short branches really are maximally exposed to the immune system, and these patients seem to do very well after surgery and rarely recur. In contrast, these patients with either short trunks and short branches, or long trunks and long branches, or short trunks and long branches, don't do particularly well. And I'll show you some early data in a minute or two that expands on this. But what we're seeing in TRACERx is when we ask for immune infiltrate estimates, either by RNAseq or pathology calls of T cells, we're seeing a relationship between, albeit relatively weak, but still there, I think, in the small cohorts, in lung adenocarcinoma, that the clonal neoantigen burden, so the number of clonal neoantigens present in the trunk of the tumour's evolutionary tree, seems to be related to the degree of T cell infiltrate we see in these tumours. So again, emphasizing the potential possibility that these clonal neoantigens are providing a sort of an alert to the immune system and attracting the immune cells. And so you'll all be familiar with the checkpoint axis of Honjo and Jim Allison, who won the Nobel Prize about 6 weeks ago. The anti-PD-1, anti-PD-L1 and CTLA-4 cascades that essentially anesthetize T cells and prevent them from reacting against neoantigens, at least in the context of tumor immune surveillance. And I mention these because in the context of anti-PD-1 and anti-CTLA-4 treated non-small cell lung cancer and melanoma, and this has been validated by Elie Van Allen's group independently now, is that the clonal neoantigen repertoire seems to be important predictor guiding response to anti-PD-1 therapy. So tumours that are relatively homogeneous and have fewer branches but long trunks, patients seem to do better on anti-PD-1 therapy and have responded by 6 months of therapy. In contrast, tumours that are more heterogeneous and have longer branches or perhaps shorter trunks in general don't do so well. Perhaps because these mutations are in some cells but not others and are less efficiently recognized by the immune system. So the question is, how are these branch neoantigens generated in melanoma? And this is a cohort of patients treated with anti-CTLA-4, a small cohort of patients with melanoma treated with anti-CTLA-4 ipilimumab. And here, similarly, we're seeing a trend where the responders have a clonal neoantigen burden and the non-responders have a tendency to have more branch mutations. And these very high neoantigen burden tumours with lots of red mutations, these are the subclonal branch mutations. Interestingly, these branch mutations all derive, these subclonal mutations, all derive from, well, many of them derive at least, from an alkylating agent signature. This is DTIC, temozolomide, alkylating agent signature. All three of these patients were treated with DTIC or temozolomide, and we suspect that led perhaps to a transient response and the selection of a subclone that had escaped treatment that now harboured many of these alkylating agent signature mutations, creating these branch mutations, if you like, cytotoxic agents, creating these branch mutations that we think are not the optimal targets for the immune response and immune surveillance in non-small cell lung cancer and perhaps melanoma. So what is the optimal neoantigen? Samra and Kevin have been working on this in the lab. And about a year ago, they published this work showing they came up with this idea that, it's pretty obvious and actually wasn't a totally novel idea and had been alluded to in the literature, but tested this in terms of checkpoint inhibitor responding patients and non-responding patients and cytolytic activity in the context of kidney cancer, with a hypothesis that an insertion-deletion frameshift mutation would create a neo-orf that would generate, actually in TRACERx, on average about 15 new amino acids that the body hasn't been exposed to or seen before, and might act as a very potent tumour vaccine potentially. And that's what we see in the context of TCGA and TRACERx data, that we see that tumours in renal TRACERx that have a high frameshift neoantigen burden also have a high cytolytic activity with T-cell gamma interferon response and cytolytic activity that we don't see for non-synonymous mutation burden. So at least in kidney cancer, it seems that it's the frameshift insertion deletion burden that seems to be best predictive of the degree of cytotoxic infiltrate and activity we see in those tumours. And when they applied the frameshift insertion deletion burden to that same cohort that I showed you earlier of anti-CTLA-4 and anti-PD-1 treated patients, it seemed to be as good a predictor of response if you had a high insertion deletion frameshift burden, as good or a better response predictor than tumour mutational burden. We're just looking at single nucleotide variants. And I guess that's not a surprise because, as I said, you've got many more mutations, many more amino acid changes that the body hasn't been exposed to before. So this talk is very neoantigen focused, and you'll forgive me for that as a non-immunologist. But one of the puzzling questions that we keep asking ourselves is, if these neoantigens are so important, why can we even detect them in these tumours? So if they're potently suppressing tumour progression, which I'm perhaps telling you they are, why are they detectable at all? So this is a question that Nikki and Rachel have been looking at in the lab, and comes back to a fascinating statistician and very, very accomplished statistician actually in the Second World War called Abraham Wald, a Jewish emigre who fled to the US and became a very central member of what was called the SRG, or the Statistical Research Group. And they were presented with this problem where they had these B-52 and Lancaster bombers coming back from Germany, shot to pieces in the fuselage, but very few bullets were present in the engines. And the RAF and the US Air Force were arguing, well, it's very clear what we need to do. We need to be strengthening the fuselage because that's where all the bullets are and that's where the maximal point of danger is. And of course, every time you strengthen a plane with armour, you make it heavier, make it harder to turn, harder to manoeuvre, and heavier on fuel, and its range decreases. So you've got to be parsimonious with your armour. Abraham Wald recognised that this was complete nonsense, and actually what you were seeing was purifying selection. What you're simply seeing, you just don't see the planes come back with bullet holes in the engines because they don't tolerate it. And they burst into flames and crash. And so actually when he measured this, the number of bullet holes per square foot, you can see the bullet holes per square foot in the engine was significantly less. I mean, you haven't got p-values here, but trust me on this, than the number of bullet holes per square foot in the fuselage. So why am I telling you this? Well, I'm telling you this because it's this concept of purifying selection I think is so important and so hard to measure in cancer. But we're beginning to see some evidence actually that there is purifying selection in the context of lung cancer evolution. That we're seeing purifying selection of ancestral clones harboring neoantigens in lung cancer genes that are generally expressed. So lung cancer expressed genes are less likely to harbour neoantigens than genes that are never expressed in lung cancer. And that may seem, it seemed to me to be a bit heretical, but it's what we're seeing in the context of TRACERx, that actually mutations in genes that are expressed in lung cancer are selected against during evolution. I guess that's obvious if I explain it like that subclones are being selected out by virtue of the fact that they're expressing mutations that appear to the immune system as being non-self. And what you're left with is essentially a fossil record of mutations in non-expressed genes in the cancer genome. And that predominantly is what we see, and indeed about 40% of clonal neoantigens are just not expressed at all, and many of those are in genes that are never expressed in lung cancer. So purifying selection. So when thinking about our brave new world of targeted neoantigen therapies, we want to be targeting multiple neoantigens. We want to be finding frame shift insertion deletion antigens that are expressed and evade nonsense-mediated decay and basically are expressed at high levels, and ideally are in genes that the tumour cannot possibly lose and has to maintain for housekeeping or for its life history in general and proliferation. And of course, that's the holy grail, and some patients may be lucky and have those, and other patients may not be. So we've now got a system in place, a sort of commercial academic enterprise, where we've got multi-region sequencing in the lab set up, where with approaches like Sine Hadrup's tetramer approach, where we can fish out, tetramers are sort of, I think of them as sort of synthetic antibodies that can sort of bind and fish out T cells. They're actually synthetic MHC complexes with a peptide that the T cell receptor can recognize and we can identify through FACS analyses. And we can now, in patients' own tumours, recognize multiple T cell clones that recognize multiple truncal alterations in individual tumours. And we've really been pleasantly surprised quite how frequent these are in patients' tumours. And I guess it's just like looking for a needle in a haystack. We need to identify them, we need to expand them, and we basically need to grow them up in overwhelming numbers and give them back to patients all within a 12-week time frame. And that's what Achilles Therapeutics is trying to do in the context of the Wellcome Trust and CRUK and the CRIC. And we hope to have a first-in-man trial set up from June next year in melanoma and lung cancer doing just that. So of course, when I first told the director of our institute this is what we were going to do, he said to me, "Charlie, do you really think we're going to beat 4 billion years of evolution in a 20-year career?" And I said, well, probably not, but we're going to give it a good shot. And since then, we've been sort of really trying to address that question, asking how might a tumour escape a clonal neoantigen therapy, and how might we be able to box it in and prevent immune escape? So in the last 5 or so minutes, I'll just quickly show some new data that's really very rough around the edges, but hopefully you'll get the idea of where we're heading. So many of you will be familiar with the sort of the first generation of tumour microenvironment analysis. This is sort of bulk RNA-seq analysis to deconvolve the tumour microenvironment to infer macrophages and neutrophils and CD4 cells, CD8 cells, etc., etc. It's pretty crude. You lose all the spatial resolution, and arguably these tools aren't very accurate themselves. And so we were aware of that, and so we went back to the bench, if you like, and said, well, how could we identify the best signature that exists currently as a sort of a skeleton to build new signatures from? And so Rachel Rosenthal had these 3 criteria by which the published gene sets had to conform to, that immune genes should not be influenced by their DNA copy number in tumours, in tumour cells. The expression of these immune genes should negatively correlate with tumour purity. In other words, the more tumour cells you have, the fewer immune cells you should have, and that immune signal should drop. And the expression of these immune genes that infer, let's say, CD8 cells or T cells should correlate with ground truth measured scored by an expert pathologist used to scoring T cells. And we've got 2 pathologists who work with our group, David Moore and Roberto Salgado, that have been doing this for many years and are very well known for their efforts in this area. And by doing this, we found that of the signatures we put through this pipeline, the so-called Danaher signature came up as being the best in terms of all those 3 criteria and the relationships to ground truth measures. So at least we've got a signature that's now benchmarked against a pathologist that's reasonable. It's not outstanding, but it's reasonable. And of course, we're aiming to go much further than this in the future, looking at single cell resolution, spatial resolution, using techniques like Codex and CyTOF and what have you. But for now, this is all we've got. So we profiled by RNA-seq about 170 tumour regions and 64 patients. Those are the tumours we had decent RNA data for. And when we do this, we find a sort of mixture of infiltrates. We see some tumours with low infiltrate, we see some tumours with high infiltrates, and we see some tumours that are sort of the straddle the middle, where some regions are high and some regions are low immune infiltration. And we can start asking some questions about why this might be, what the mechanisms of immune escape might be within those individual tumour regions, and how they correlate with whether those tumour regions are highly infiltrated with T cells or not infiltrated with T cells at all, and why we see mixtures. And first of all, what we're beginning to see is a hint that sort of validates prior work from Saurabh Shah and others in Memorial Sloan Kettering, that tumours that have high immune infiltrate tend to have lower diversity, suggesting perhaps, I don't know, but that one explanation might be that the immune cells themselves are pruning the evolutionary trees and reducing diversification. They're pruning off those branches, pruning off those subclones as they evolve. So there are an extensive list, we think, of immune evasion mechanisms in lung cancer, and we think we're beginning to see some early insights into how they may segregate between low infiltrated tumours and highly infiltrated tumours. So the first model I want to suggest to you is that chromosomal instability is important in this. And it's important because chromosomal instability does something that doesn't happen in sexually reproducing organisms by and large, that it can remodel phylogenetic ancestries. And it does this in this thought experiment as follows. So let's say you've got an early mutation that occurs in the trunk as the tumour is evolving. On this, let's say chromosome 18q here, on the paternal allele. That could be a very potent neoantigen thought experiment. As a tumour evolves and gets subclones, still truncal obviously, what you would have thought once truncal, always truncal, but actually what might happen is over time, if that is a very potent neoantigen, there may be selection for loss of chromosome 18q, which encodes that potent neoantigen. And so that previously clonal neoantigen may now become subclonal. There is a sort of illusion of heterogeneity in a way, that we may see that some heterogeneous mutations are heterogeneous, but once upon a time in evolutionary history were once clonal. And we do in fact see this quite frequently. It turns out that about 15% of heterogeneous mutations we predict from our multi-region data, looking at chromosome copy number events, result from loss of one chromosome arm or one small chromosome segment in one small subclone of that tumour. So that branch mutation really isn't branched at all. It was truncal, but has now become branched. And the question is why? So we think one reason might be immune evasion. So in low immune infiltrate tumours, what we're seeing is evidence that in the low immune infiltrate tumours, there's an increased frequency of copy number loss of neoantigens. that were likely to have been clonal in ancestral time. So maybe these tumours were highly infiltrated once upon a time. They were subject to selection through loss of clonal neoantigens, and they no longer have a signal to the immune system perhaps, and the T cells disappear. That's the hypothesis, and we're going to start to test that in mouse models. So what about the highly immune infiltrated tumours? So in these tumours, I mentioned to you earlier, they're enriched for HLA loss of heterozygosity. So this is looking now at lung adeno and lung squamous tumours across those 100 patients and about 300 tumour regions. Richly infiltrated, homogeneously infiltrated, high tumours with lots of immune infiltrate, mixed tumours and sparsely infiltrated tumours. And what you can begin to see here is the mutual exclusivity of HLA loss of heterozygosity for mutations in the antigen presentation machinery. So regions of the tumour that are hot and diffusely infiltrated with immune cells seem to either lose HLA or mutate their antigen presentation machinery. And some tumours can do both, depending on which region you're looking at. But you don't, I say both. So one region might have HLA loss of heterozygosity, another region may have antigen presentation and machinery mutations. And it's less common to have both. I mean, there are some tumours that have both, but very often these are mutually exclusive events, as you might expect. And we see also in these highly infiltrated tumours, we see evidence of neoantigen transcriptional depletion. So where these neoantigens do occur in genes expressed in lung cancer, very often we find in tumour regions that are still intact for HLA, and that's important because we don't see this signal in tumour regions that aren't that have HLA loss, that have HLA loss of heterozygosity. We see evidence of transcriptional repression of those neoantigens. So those neoantigens just aren't expressed at all. So obviously the question is why, and so we turn to Stefan and Beck and Peter van Loo's group, who've been profiling these tumours through RBBS methylation profiling. And what we find is that these neoantigens in these hot regions with intact HLA have a tendency to repress those neoantigens by hypermethylation of the promoter regions. So these non-expressed neoantigen promoters are much more likely, we think, to be hypermethylated through promoter methylation than expressed neoantigens, and that's work from Peter van Loo and Stefan Beck's group. So in my last minute, I'm just going to say tentatively what we think this might have in terms of impact on clinical outcome. And again, take this with a pinch of salt. This is early data, discovery data, and we've got this is only data in the first 100 patients, and we're going to be looking, obviously, at 600 or 700 patients over the course of the next 18 months to see if this validates. But we wondered whether there would be a difference in terms of outcome, in terms of low immune evasion tumours versus high immune evasion tumours. I'm sorry about this rather childish cartoon I put together here, but it helps get the message across in a short time. So let's say you have a tumour with high immune evasion capacity. So these will be tumours with a low immune infiltrate or evidence of the immune escape mechanisms I've shown you in the last few slides. In contrast, what about tumours with low immune evasion capacity? So these may be tumours with high immune infiltration or no evidence of immune escape. And when we put that to the test in relapse-free survival terms in the TRACERx adjuvant cohort, we see that tumours with low immune evasion capacity do better than those with high immune evasion capacity in this analysis. I mentioned earlier that tumours with high clonal neoantigen burden do very well and rarely relapse. So let's take those out of the mix now, and let's look specifically at the patients with low clonal neoantigen burden in the lower 3 quartiles. Are there some patients with lower clonal neoantigen burden tumours that still do well? And it turns out we think there are, and those are the tumours that specifically have low immune evasion. So these are tumours that have high immune infiltrate and no evidence of immune escape in terms of antigen presentation machinery dysfunction, HLA loss of heterozygosity, or other evidence of immune editing. And these patients, despite the fact they have shorter trunks, still seem to do quite well because they haven't lost the ability to have their tumours surveyed adequately by the immune system. So in summary, my last, I guess, cartoon slide before I summarise is this, that you have, we think the model we're working towards is that you've got low immune infiltrate tumours that often have low clonal neoantigen burden, and perhaps they're low clonal neoantigen burden because they've lost neoantigens in evolutionary time through copy number loss of those clonal neoantigens. And then you've had high immune infiltrate tumours, and these are enriched for tumours with high clonal neoantigen burden that may act as a sort of signal for immune recognition. We don't know, but it's just a thought, that are struggling in this sort of evolutionary predator-prey tension between the immune system and the tumour. And what's happening is we're seeing evidence of transcriptional neoantigen depletion through methylation of promoters, HLA loss of heterozygosity, and mutations in the antigen presentation machinery. And we're beginning to see evidence, at least early evidence in a discovery cohort, this pans out in survival analysis where tumours with high immune infiltrate and low immune evasion capacity do better than tumours with high immune evasion and low clonal neoantigen burden. So there are many more questions than answers, and one of the awful things about this talk that I give is I'm hopeless at answering questions, because this just opens up a Pandora box of questions we don't have answers to at the moment, but certainly enough work to keep us busy for a decade or more. But we don't know anything about the transient nature of this RNA transcript depletion, and there is this intriguing data coming out of the clinic now that if you expose a patient to pembrolizumab and they get acquired resistance to therapy, and then sometime later on re-challenge them, patients who've previously responded, there's up to a 40% response rate in those patients. And the hypothesis we have at the moment is maybe this is due to the fact you've got these expressed neoantigens in genes that are essential for lung cancer proliferation and fitness that are transiently repressed by methylation, that have got neoantigens in, just happen to have neoantigens in, that you select for repression of those by methylation that's transient, and that when you release the patient from the drug, you can get de-repression of those promoters. Those neoantigens are re-expressed, and you go in again with a checkpoint inhibitor therapy, and the patient re-responds. That's my hypothesis. It could be complete nonsense, but that's something we want to put to the test in our Darwin program of clinical trials in TRACERx. We don't know how frequently the depletion of these transcripts is acquired. We want to look over many more patients. We've got no idea what treatment does. We've got no idea what impact any of this has to immunotherapy by and large. And whether this remains prognostic in the context of the modern era of immunotherapy approaches. But I remain optimistic that adoptive cell therapies targeted against clonal frameshift neoantigens that evade nonsense-mediated decay may be really very exciting, particularly if we're going for genes that are expressed and have to be expressed in lung cancer genomes. I've said to you that 40% of clonal neoantigens are not expressed, and there is a sort of purifying selection for neoantigens in non-expressed genes that we need to be acutely aware of in any neoantigen pipeline. And I guess the key question is, can these neoantigens be prioritized? For instance, based on essential genes in regions of loss of heterozygosity. Because if you've lost a wild-type gene and all you've got left is a single point mutation that acts as a potent neoantigen, the tumor can't lose that now because it's got no other copy to fall back on. And so we could potentially target those neoantigens mercilessly, and that's what we hope to do. So we're trying to integrate genomics with cancer immunotherapy. We're very much a novice in the latter, and we're learning with the former. But I remain naively optimistic that knowledge is power, and that we may be able to make some inroads in the future for our patients. And so chromosomal instability and complexity, I hope I've convinced you, is much more than a bystander. This rewires tumour genomes, it rewires evolutionary histories, has a panoply of effects, we think, in terms of HLA loss, in terms of neoantigen deletion, in terms of parallel evolution, and selection of high-risk copy number events at the metastatic transition, which I haven't had time to tell you. I'm not going to reiterate everything I've just said, other than to say that we'll be starting first-in-human trials in the middle of next year, where we'll put all of this to the test, and I hope we'll be able to make some inroads. And so I just want to thank the lab, old and new. I couldn't have done any of this without the past members of the lab over the last decade who've just been absolutely fabulous and been a real pleasure and joy to work with. It really is all their work, this. I just come here and tell you about it. And this is all of our funders, the patients and the families have just been amazing. And Cancer Research UK, the ERC and the Royal Society and the CRUK for supporting this work. And Simon, and to you all, thank you very much for inviting me.